molecular biology that occurred in the middle of the century, from the 1950s into the 60s, and really up to about 1970 or so, we came to a conceptual understanding that genes encode proteins, and therefore these two different ways of looking at the organism, organism minus a component, components minus an organism, were complementary points of view. And in theory, you could go from a gene sequence to a protein sequence back to a gene sequence. You could go from a gene sequence to its function to a protein, except for one tiny detail. This was all just conceptual. Conceptually, we understood by about 1970 that the DNA made the RNA made the protein carried out the function. But as of then, you couldn't individually work with or purify the DNA corresponding to any particular gene. All of the inferences had been indirect inferences. Indirect inferences from bacterial genetics and bacterial regulation, or Mendelssohn-Stahl experiments, and all sorts of interesting indirect ways working out the genetic code, but it didn't let you read anything. This was a problem. Some people in the late 1960s said, great, molecular biology is over, we understand in principle how life works, now let's go understand how the brain works. And there was an exodus of some people from molecular biology into neurobiology to now go nail the brain. Figured that would be worth another 10 years or so. But which is floating around in the cell, accidentally, quote in quotes, methylated the virus's DNA. What would happen then? The virus is optimized than we are. And so you go ask and say, has any bacteria worked out how to do this? Turns out bacteria have worked out how to do this just fine. In fact, most bacteria, at least many bacteria, contain within them, in addition to their own chromosome, small circles of DNA. These are called episomes. This is the chromosome. Epi means on top of, or in addition to. So in addition to the chromosome, there's an episome. The episome is in fact an autonomously replicating piece of DNA that has an origin. And it replicates. Why do bacteria have episomes? Turns out episomes, often encoded on it, an enzyme that can say break down ampicillin. And what do I do to my Petri plate? I just add ampicillin. Now, even though most of the bacteria have not picked up a plasmid, only those bacteria that have picked up a plasmid have the ampicillin resistance gene and can grow on an ampicillin-containing plate. Now, how do I get a plasmid with an ampicillin resistance gene? It's in the catalog. It's all there, right? In fact, these occur naturally. You can, with restriction enzymes, move the ampicillin resistance gene to your favorite plasmid. If you don't like that, you could put in kanamycin resistance, et cetera, et cetera, et cetera. So that's how you do it. So we've got the big picture here. We have now gotten a library. The library of human fragments. Contained in E. coli. The library, it's a big Petri plate or many Petri plates, each one of which is a colony. Each colony has a single